Continuing where we left off, the last section we were looking at transport of materials through the cell membrane. And we started off by kind of looking at the nature of the membrane and what kinds of things would influence that transport or that movement of things either in or out of the cell. Got a little bit cut off though, so I'm going to back up just a bit and we're going to take a look at this idea of tonicity. So tonicity has to do with how you might think of it as actually sort of like toned the cell is, and it's going to relate to how the volume of the cell is because of the fluids. So thinking about the volume, I'm trying to move this over just a little bit if I can. The word at the top is tonicity. So the solutions can come in and out of the cell, and that's going to depend on the things which are actually dissolved in them, as well as the concentration gradients on either side of the membrane. So we're going to look at this again, this idea that cells have there's different possibilities for what's going to happen to them depending on the solutions that they are suspended in or surrounded by. The first one we looked at is the red blood cell example. So we were looking at animal cells, cells that don't have any exterior support like a cell wall, and the fact that they have their own internal concentration of different solutes dissolved in solution. And if they're in a solution that's on the outside that's similar to what their solute concentration of the solutions are on the interior, then we say they're in an isotonic solution. If they're in something that has a lot of solutes dissolved in it out on the outside relative to the cell itself, then it's in a hypertonic solution. Hyper meaning like there's lots of solutes dissolved on the outside, on this fluid on the outside. So the tendency is for water to move out of the cell and into that hypertonic solution. The opposite is true if it's hypotonic. That means that there's actually more solutes within the cell than there is in the solution that's surrounding it, and so water will tend to go in. Now, in a plant, we have the advantage, or a plant does, of having a wall. And that wall gives some pushback against certain conditions. So in an isotonic solution, here you can see the vacuole. We see that there's kind of an even exchange, just like in the animal cell, of water coming in and going out. However, one thing I didn't note on the last video is that notice what's happening here to the cell membrane. So the cell membrane normally is right up against this cell wall, and look where it is. It's actually pulled in just a little bit, which basically is just really indicating that this is could be a little bit more filled in the inside. So cells, plant cells, actually like hypotonic solutions because that allows their vacuole to fully fill like a balloon that you've blown up to its maximum. And that allows for the cell membrane to be now flush against this cell wall. And it gives some internal pressure that supports the cell. And the opposite condition, if the cells have a hypertonic condition where there's more like solutes and stuff dissolved outside than inside the cells, then water moves out of the cell. And look what happens. The central vacuole shrinks up, and even more than that, the membrane shrinks in. So that when the plasma membrane shrinks in, that's called plasmolysis, and that's what you can see here. So without adequate water, you can see what happens is your plant wilts. So when your plant is wilted, it just means that the pressure on the inside of those cells is not sufficient. And so in order to do that, we need to water the plant. And so you can see on the right, the plant has a lot of water. It's nice and upright. It's not falling again. It's not like wilting against the stem or any of that. And so what this is showing is something called turgor pressure. Turgor pressure is the pressure from the interior of the plant cell. So coming back here, there's high turgor pressure in this hypotonic cell. average in the isotonic and very low on the hypertonic. Animals, especially single-celled organisms, also have to have a mechanism for controlling the amount of water, especially ones that live in water. So they're always trying to control how much water can come in, and in order that they don't explode, they don't burst open. So they have, many of them, a little device, which is a vacuole, but this vacuole actually can pump. It can remove water. So it controls when water starts coming into the cell. So if this little guy started swimming into an area that had a lot more fresh water and less salt water, then water would start to flow in. So it can pump it back out. It can control that. So that's kind of a cool little feature. Other organisms, such as the ones that live in salt water, may be able to remove salt from their bodies, so they can reduce the amount of salt that they have in them. Fish, you can see what they do. They basically excrete a very dilute urine. And then the brain actually has these things called osmo referring to water, and you'll see why coming up, receptors. So osmoreceptors in the brain they actually monitor this whole condition, but in us, to say, well, how's the fluid balance in our body? And do we need more? Do we need to drink water? Or do we need to release water in the form of urine? And so let's activate the kidneys. Active transport is the movement of materials in or out of a cell, but instead of it flowing in the direction of the concentration gradient, it flows the opposite. It flows against its concentration gradient. So let's say a cell has a lot of glucose in it, and they give this as an example above, but the cell still wants to take in more glucose. So normally if there's a lot of glucose in the cell relative to out of the cell, then the opposite would happen. This glucose might want to go back out, and the glucose that's on the outside in the bloodstream wouldn't want to come in. But a cell has a way of getting around this and using energy to move it in the opposite direction of the way it would normally flow. So it'll be against its concentration gradient, going from a low concentration into an area where there's a higher concentration of that material. And we'll look at some specific examples of those coming up. Another possible way that we have to consider is not just in terms of the amount of a solute, like how much glucose there is, that concentration of that material, but also where are the charges. So in some materials, like ions, the charge could be an issue because sometimes the positive charges want to go in, because there's more negative charges inside the cell, and therefore sometimes that's not a desirable thing. And so, and I'll explain this a little bit more in my notability lecture piece. So the active transport can be used to maintain, in this case, an imbalance, trying to keep certain ions on one side or the other of the membrane. There are two main types of active transport. The primary type is where that molecule, that energy molecule we mentioned before, ATP, is going to be used directly. So it's like we're plugging in something, we're lighting something on fire, we're using energy for a purpose. But there's another type of active transport that's called secondary. And secondary active transport basically is using the power of primary active transport, but then using that as a way of moving something else. And let's look at a real example here in a few minutes. So these are electrochemical gradients. An electrochemical gradient has to do with where the charges are on one side of the membrane or the other. And these gradients, so when you think gradient, you want to think of it's usually not balanced. It's not equilibrium. It's like one side has more of something than the other. But we're looking here at electrochemical gradients, so we're kind of talking about both here. Concentration of something and what those or what some particles might have in terms of their charge, if they have a positive or negative charge. So an electrical gradient basically is something that's actually a good thing for the cell. The cell wants to create an electrical gradient. And what it wants, in general, is to have a certain charge on one side of the membrane. So when the cytoplasm is going to contain more negatively charged molecules, right, and that's actually what it wants. It wants to have more negative charges on the inside. So that means on the extracellular side, on the outside, I'm doing this opposite because I can't see here, that the outside of it generally has a higher concentration of positive charges. So the cell is going to use energy to maintain that imbalance of having certain charges on the inside and on the outside. Now on this picture, what they're showing you, actually the next picture shows a little better, so I'll go there. So, because it's showing what's happening here. So if the net charge on the here we go, extracellular side, so here we are outside in the cytoplasm, we have a lot of sodiums here, and then we have some other positively charged ions like potassium as well, but then we have negative charges, so here's the chloride ions here. If we want to maintain a certain imbalance, we're going to move the charges to the outside that we want to keep, and we want to keep a higher positive charge on the outside. And we're going to try to maintain a higher negative charge on the inside. Now, one of the ways the negative charge is maintained, though, is that there's some large negatively charged amino acids inside the cytoplasm that are kind of keeping a higher negative charge here, and then we're trying to control the direction of flow of any of the positive charges. So again, I'll show you this a little bit more on notability as we go forward. Another thing that happens is we have to look not at just the flow of the individual particles, but we also want to see what's the role of those carrier proteins, the proteins that are in the membrane itself. So some carrier proteins will move something just in one direction, and that's called a uniporter. And it's going to carry just one, well, I shouldn't say just one direction. It's going to, one molecule or one ion, it's going to move one way or the other. But it's kind of like, it's in charge of a particular ion or particular molecule. A symporter is one that can bring in or out two different things, and they go in the same direction. So sim is like in similar, they're going the same way. And then there's one that's called an antiporter, which can bring one thing in while it takes another thing out. Here's an example of a very important active transport mechanism in all cells. Remember, we're looking here at primary active transport, meaning we're using energy, we're using ATP. And we're going to take ATP actually and break it down. That's why this word hydrolysis is here. So the breakdown of ATP by hydrolysis is going to release the energy that's needed to move whatever we're trying to move in or out. And the most famous example of this is the sodium-potassium pump. So it's the sodium-potassium pump that's going to move sodium out. It's going to bring potassium in. Notice that the ratio, though, is more sodium are going to go out than potassium are going to be coming in. So the question on this is the pump uniporter, symporter, or antiporter? And this is also called an electrogenic pump, which I want you to think about what that means. We'll talk about that.